Good morning, good afternoon, everybody. I'm Mike Rice from Cello Health Bioconsulting. I lead the advanced therapeutics practice, which deals with advances in technological platforms, anywhere from biologics, nucleic acids, cell and gene therapies. We have a great panel to share with you today, Investing in Biologics panel, that will talk about different aspects of the dynamics of the biologics markets, as well as how different technology platforms are advancing access to new targets, new indications, improving upon validated targets, and other aspects of addressing unmet needs in the oncology market. So as you know, and you may have seen the Nature paper that came out last week or a couple weeks ago, talking about the landmark of the 100th monoclonal antibody that was approved by the FDA. 41 of them are oncology products. So there are many of them have been approved outside of oncology, but certainly oncology is a leading area for progress and innovation in the space. The oncology market is now exceeding, the oncology monoclonal antibody market is now exceeding $60 billion. However, the leading brands, the most validated tumor-associated antigens that have been sought after and developed over the years are facing biosimilar competition. So that's pushing the pressures on target product profiles needed to be achieved to be competitive in those spaces. And so, it'd be great to hear the panelists' perception of that, going forward. However, even though we know it won't play out like a generic market, we expect the biologic markets to monoclonal antibody market, maybe not even including therapeutic proteins, to double in the next 5 years. So significant growth, expanding to new novel targets, looking at life cycle management strategies, including antibody drug conjugates, FC engineering, other things to push the envelope on those particular validated targets. And then we're looking at a plethora of new targets coming in. And then what is really exciting, what's going on is really the innovation in multifunctional, bifunctional, targeted cytokines, shielded cytokines, ways to engage dual targets, ways to engage multifunctions into one antibody product that are really pushing the capabilities. And it'll be interesting to see the way that this plays out because, as you know, the adoptive cellular therapy space has also enjoyed incredible investment into that. Much of the more recent investments have been driving more off-the-shelf allogeneic products. Some of them ultimately could be completely off-the-shelf, like a highly edited iPSC product that, at least in the near term, is positioned to be a short duration treatment and have retreatment such as we see with antibodies and things like that. So there'll be a really interesting competitive dynamics. At the same time, I think a lot of the panelists today will be saying, well, we can play with adoptive cellular therapies such as NK cells, and we can also engineer things into our biologics that obviate the need for cells and engage endogenous cells and things like that. So be great to hear the panel's discussion of all this dynamics, their position in this field, hear a little bit aspects of how the investors are seeing the balance of all these different modalities, and look for an enjoyable discussion. We're going to kick it off with a round of introductions where everybody can give a little bit of a overview of their technology platform, what they're doing, and how they see developing further, as well as anything that's going on in financing or partnerships. So let's try to maybe go down alphabetical order. So Lisa Decker from IGM Biosciences, do you want to introduce yourself and talk a little bit about what you're doing?
Absolutely. Thanks, Mike. Hello, everybody. Really excited to be a part of the panel, and thanks for including me. My name is Lisa Decker. I joined IGM just about 2 months ago now as Chief Business Officer. IGM Biosciences is a clinical stage biotech company located in Mountain View, California. And maybe as our name suggests, we are focused on the development of engineered IgM antibodies. Our platform is focused right now on oncology, although we have the ability to apply the platform to autoimmunity and infectious disease as well. We are building out a very broad pipeline within the oncology space, and it encompasses right now 3 main areas. First, on T-cell engaging bispecifics. Next, on really leveraging the ability to cross-link agonists, most particularly TNF receptor superfamily members. And then the third aspect, and the one that you touched upon, Mike, is on targeted cytokines. We have 2 programs in the clinic right now, with a third expected by the end of the year. Our most advanced program is a CD20 by CD3 bispecific in development for NHL malignancies. Following that, also in the clinic, is our DR5 agonist program, which is in development for both solid and heme malignancies. And we expect by the end of the year to have an IND filed on our targeted cytokine, which is a PD-L1 by IL-15 bispecific.
Great, thanks, Lisa. Kris Elverum from Rubius Therapeutics. Would you like to go next?
Sure. Thanks, Mike, and thanks for including me in the panel. So I'm Kris Elverum, I lead business development and strategy for Rubius. Rubius is the leading company that's engineering red blood cells as therapeutics. We have a unique platform that allows us to engineer red blood cells with target proteins of interest either on the surface of those cells, across the membrane, or inside of the red cells. And we think that that has many advantages. I think first, in terms of potency, the cellular presentation of desired cytokines or other agonists, and oncology in particular, we think is a more potent approach to engaging the immune system, taking advantage of direct cell-to-cell interactions. But then as well, the second advantage we think is safety, in that our red cells are confined to the vasculature. So much of the mechanism that has held back other pathways like 4-1BB, we believe that we will avoid, and our early clinical data shows that we have an advantage in terms of the tolerability of at least our initial clinical data. And then finally, in terms of scalability, red cells truly are off the shelf. They've probably the first cell therapy in terms of red blood cell transfusions have been given to people for a very long time. And we make our cells in bioreactors in a highly scalable way that we believe long-term will be able to be delivered to patients much in the same way that monoclonal antibodies could be. In terms of our early data, we released in March our first clinical data for our lead program, which is a red cell that has 2 agonists on the surface, a 4-1BB ligand in its natural trimer form, as well as an IL-15 trans presentation. And that early clinical data was quite exciting. It's the first 14 patients in dose escalation, but we did observe 2 partial responses and a very clean safety profile, which notably is quite different than what you've seen in other agonist approaches. So looking forward to the rest of the discussion, Mike, and I'll turn it back to you.
Thanks, Kris. So Elliot Foster from F-Star Biotechnologies.
Hey, Mike, thanks. And fellow panel members, delighted to be here. Elliot Foster, I'm CEO at F-Star. We're another bispecific firm. We've been around for quite a long time, around a decade, based on a kind of breakthrough observation that if you take a fully human IgG1 and make a few handfuls of substitutions in the terminus of the FC region of that antibody, you can create a 2+2 bispecific. So retaining all of the advantages of a natural human format with respect to bringing cells together and clustering the receptors one's targeting, but at the same time, easy to manufacture, low immunogenicity, and so on. We've got 4 programs in the clinic. We listed on NASDAQ in November of last year, and we closed a financing a couple of weeks ago for 65 million in a in a confidentially offered financing, which was underwritten by SVB Leerink, which was great. And that gives us a good runway for a couple of years. The lead programs go after both agonists and antagonists. So our most advanced program is in phase 2 with a LAG3 PD-L1 bispecific, LAG3 becoming very popular given phase 3 pivotal data being announced by BMS, and we're all very excited to see what comes out of ASCO in a couple of weeks. A T-cell redirector, which is PD-L1 and CD137 or 41BB. And what we do to avoid that kind of first generation is silence the Fc gamma receptor to avoid ADCC, so avoid killing the cells we stimulated, and that has demonstrated very nice safety and tolerability. We also have a first-in-class dual agonist, which is an OX40 CD137, again in phase 1, and very excited about that. And then kind of left field, we have a second-gen STING agonist, and very excited to report data on that in the next couple of months or so. So delighted to be here, looking forward to the discussion. Thanks, Mike.
Thanks, Elliot. So Rachael Lester from Harpoon Therapeutics.
Good morning, everyone. Nice to see this panel again. We had a very lively discussion pulling into this meeting, and so looking forward to today's discussion. Thanks for allowing me to be here, Mike. So my name is Rachael Lester. I head up corporate strategy and business development for Harpoon Therapeutics. And Harpoon is focused really on very specific vertical within the bispecific field, which is T-cell engagers. So we focus specifically on the T-cell as an effector cell. And we like to believe that we are standing on the shoulders of giants, so to speak, when you think about some of the history and what's been done with T-cell engagers in the past, as well as what's been demonstrated with CAR-Ts and the utility of these programs, specifically in heme malignancies. And what we've tried to do is take that challenge to the next level into solid tumors. And the way we've done that is we focus on addressing a few of the challenges in this field, initially around stability and half-life extension. So the first generation BiTE therapies, which we know were initiated by Micromet and then acquired by Amgen, have to be delivered by continuous IV. And so half-life was a big part of this story. Many of our brethren, and maybe some on this panel, have used antibodies and Fc domain as a way of extending half-life. We have used a technique using albumin, which was, I think, initially pioneered by Ablinks to extend half-life. And we think this will give us additional stability and safety window for these types of therapeutics, and offering, in contrast to the CAR-Ts, an off-the-shelf method. So our first and a foundational platform is called TriTAC, a tri-specific T-cell activating construct. And these are built using single domain antibodies and built to keep the molecule both small, globular, and flexible for optimal penetration into solid tumors. Mike, you mentioned in your intro the idea of shielded cytokines or shielded effector cells. Our second next-gen platform is ProTriTAC. It is a prodrug format of TriTAC and essentially shields the CD3 from being activated until it is in the tumor microenvironment. And that is a platform that we debuted at SITC and provided some nice proof of concept on that platform, and looking forward to that coming forward. You may know some similar technologies. Harpoon spun out a company called Maverick several years ago, which was focused on a very similar concept, as well as a company called Werewolf, which was focused on a pro format for cytokines. So this will be a very hot area, as I expect to see. Our portfolio is now, we have 5 programs that we're moving forward. Our TriTac programs are focused on PSMA for prostate cancer, mesothelin, which is a target for a variety of solid tumors, but which we are exploring in ovarian, pancreatic, and mesothelioma cancers. And we're targeting BCMA for multiple myeloma. And our last TriTac, which went into the clinic this past December, is targeting DLL3 in small cell lung cancer. Our first program and our lead program on our Pro-TriTac platform will be looking at EpCAM, and that's across a variety of solid tumors. And we're looking for that to get into the clinic, hopefully late next year. So thank you, Mike. Looking forward to the rest of the discussion.
Great, thank you. There's something floating around here. Okay, and last we have Nanna Luneborg from Novo Ventures.
Hi everyone, good to speak with everyone again. Nanna Luneborg, I'm part of Novo Ventures. Novo Ventures is part of Novo Holdings, an asset manager with more than 60 billion euros under management and an active investment portfolio around 20 billion. Novo Ventures invests across biotech and medtech, private and public, US and Europe. We have a current portfolio around 70 companies, and our current investment pace is around 500 million per year, with a ticket size in the sort of 10 to 30 million per investment. Our offices are in San Francisco, Boston, London, and Copenhagen. I'm in Copenhagen and part of the team leading our European activities. Immuno-oncology and biologics platform has been a busy area of investment for us, and I'll mention some of the companies in our current portfolio. NBE Therapeutics, which was acquired earlier this year by Boehringer Ingelheim, ADC company. Lava Therapeutics, we led the Series C investment last year, and the company went public in March of this year. Bispecific gamma delta T cell engagers. Bolt Biotherapeutics, another novel conjugation platform, also went public earlier this year. And some in the cell therapy space, Nkarta and Arcellx, and also Checkmate and IO Biotech are some other examples from our portfolio. So it's an area that we're extremely interested in, and I think a lot of the technologies being developed by some of the companies on the panel are great examples of what we'll hopefully see as next generation combinations in this area. Look forward to the discussion, and thanks for inviting me.
Great, thanks. All right, so let's kick off the discussion. And a couple of things came to thought just hearing your introductions, and thanks, Rachael, for explaining the shielded cytokine side of things. We heard a couple other things thrown out: NK engagers, T cell engagers. We also heard T cell redirectors, that there was a difference between them. I guess, I just like to start off maybe at high level general discussion around what sort of, indications and, how you've selected those indications that you have, in your pipelines, and how are your particular platform approaches, most suited for winning in those indications. Maybe, I know it's a very broad discussion, but who would like to sort of kick off that topic?
Sure, why don't I take a minute or two, and then maybe others can build on the conversation. So
you described it, the T cell redirector, I think is the term you used, and I was wondering if there's a difference between the redirection and engagement.
Yeah, so I guess anything that targets a T cell biological target is a T cell engager. So a redirector is really what bispecifics do, and I guess to some extent CAR Ts do, but it's carrying the T cell on the back of the director that's sitting within it. But so the redirectors we like to think of. So we use PD-L1, a well-known checkpoint pathway, because it's overexpressed. It blocks the checkpoint pathway as an antagonist, but is also overexpressed in cancers, in particular those that have become exhausted, so post-checkpoint therapy, for example. And so you not only get blockade of that pathway, but you also get a quasi-tumor-associated antigen. Now, many other companies actually use tumor-associated antigens as targets, and I guess the best example from CAR T is CD19, which appears in many hematological cancers, as the kind of most obvious and well-known example in that setting. And there are many of them now in the industry, both proteins and non-proteins. We particularly like PD-L1 for that reason, so that redirect. So one of our programs, FS222, binds to those overexpressing PD-L1 tumor cells and then redirects the immune cell through the CD137 or 4-1BB and many different types of T cells. I mean, not just CD8s, but 4s and NK and others into that environment. So that's really the idea of a redirection rather than just an engagement, because indeed monotherapies can work in an engaging way. And I guess, Mike, if I might steal the floor for another 30 seconds, one of the comments you made in your introduction was interesting. There are indeed 40 plus monoclonals now and other proteins approved in oncology. But of course, what's a truism in oncology is that no one uses a drug alone. In practice, there are combinations of chemo, radio, monoclonals, targeted therapies, DNA damage repair, that are on and on and on. And the fact is that all of those drugs become the armamentarium for the next generation to sit alongside of it. And all of our companies will be thinking not just about demonstrating that monotherapy, even though it's multi-specific, that monotherapy approach, but also how it's used in combination with those other drug types that exist. And so all of that really says you need to have smart development, thinking about the right patient with the right pathway, but also really understand where the market is going to go over the next 5 to 10 years, because we're going to be sitting in that environment and actually bringing better outcomes for patients, which ultimately is what we're looking for. So bringing those two things together, I couldn't help but jump in there, and I'll stop.
Yeah. Who wants to go next? Kris, you want to talk a little bit about your indication selections and why your platform's uniquely positioned for that?
Sure, happy to. Yeah, and we take a little bit of a different approach than those that are seeking to mask or activate in the tumor microenvironment. Our hypothesis is that the right immune cells to engage are the non-exhausted cells in the periphery. So our red cells express 4-1BB and IL-15 on the surface and engage T cells and red cells in the spleen and in the vasculature, and those then traffic to the tumor sites and have a killing effect within the tumor site. And we have observed a lot of trafficking into tumors of those healthy NK and T cells. And we think that we avoid some of the toxicity because our therapeutic does not go to the bone marrow or the liver or other places where a lot of tox has been observed from some of the other therapies. In terms of indication selection, Mike, we're taking a very an approach of understanding first and foremost what kind of receptors and activation markers are we seeing in the T cells and NK cells that we're engaging, and then looking for particularly for NK cells, looking for the corresponding markers in tumors. And so you observe tumor types that overexpress certain markers, and if we're activating the NK cells that would match, then those are going to be more likely to be able to be beneficial, right? So, for us, that has us, the known biology of NK cells and AML is, of course, very clear. But then as well, there's quite a few really interesting solid tumors that have shown to overexpress the desired target. So that's how we're thinking about indication selection, is really from the biology first, and then looking at how might we think about development within that biology.
Mm-hmm. Great. Lisa, do you want to talk a little bit about maybe how you're balancing the risk in indication selection and why your platform is uniquely positioned for those areas?
Yeah, happy to. Thanks for the question. So, when IGM really thinks about what indications to go after and where to apply our technology, sort of at the core of it is really focusing on the advantages that an IGM format can deliver. And there are a few that come to the top of the list, and I can give a couple of examples of how that drives maybe what we're thinking in terms of indications. So, for example, Because IgMs, by definition, have more binding sites than an IgG, we think one of the advantages of IgM is this ability really to have stronger binding to cell surface targets, including those with low expression of a particular target. So we may get more complete targeting of cancer cells as well as better binding. And one example of that is with our CD20 by CD3 bispecific program. Right now, it is underway in phase 1 in non-Hodgkin's indications. But because we think that IgMs have this ability to really, play to the strength of the avidity story and target low expressing cell types, we're looking to expand into a CLL indication, so something with a low expression of CD20. So that's just one example of how we try to make sure we harness the strengths of the IgM platform in making decisions about what types of indications that we go after. Also, because we do have this stronger binding to targets, especially difficult targets like carbohydrates, we think that the IgM format potentially opens up a new range of targets that may not have been addressable in the past. So those are just a couple of examples of how we're thinking of applying our platform.
Great. And for the audience that doesn't know what an IgM, the structure is right over Lisa's left shoulder there with 8 binding sites, I believe, right?
10 binding sites.
10 binding sites. Okay.
As compared to an IgG, which has 2.
Great. Right. Okay. So maybe shift gears a little bit. If Rachael, maybe you could kick us off a little discussion about, Kris brought up, the focus on NK cells or how they're using, biomarkers to identify the right immune cells and everything that they're trying to recruit. Maybe you could talk a little bit about, your approach and, why is, an NK cell or T cell or gamma delta T, like what is the best cell to engage and why is your platform uniquely positioned for that, if there is an answer to that?
Yeah, it's a great question. I actually think the jury's still out on that particular question, where, addressing both the question about indications as well as this one, for us, what we believe is that your T cells are pretty much the most powerful natural killer, not to use that word incorrectly, but killing cell in your body, right? And so if you can harness the power of the T cell, you should be able to eliminate the cells that are expressing your target on those targeted tumor cells. And so, for example, as we've thought about our indications and thinking about the biology first, what we really do is think about what the target and what the target expression profile looks like. And so when you think about that and you're looking for targets that are expressed more uniquely or overexpressed more uniquely on tumor cells as opposed to healthy tissue, because essentially those T cells will take out whatever tissue is expressing that target. And that's what drives not only the CRS, but some of the other toxicity associated with T cell engagers and CAR T programs, which is why we've seen a lot of initial success with our heme malignancies. These are taking out cells that essentially can be taken out without giant toxicity effects, but we have a little bit more challenge moving into solid tumors. And that's why, we have to be very, very selective about which targets we go after. And for most companies, you'll see them pursuing, I'd say there's a handful of targets that are on the top of the list for most companies with regard to T cells and solid tumors. And from that perspective, our solid tumor pipeline is not that unique. So you'll see a lot of our same targets being pursued by Amgen and others. But, you look at our second program targeting mesothelin, we've initially gone after indications where we have over 80% expression of mesothelin in those tumor types. Now, mesothelin, if proven to be, if we can achieve proof of concept there, what you would imagine is you could also see this being applicable to many other tumors, as things like triple negative breast cancer, non-small cell lung cancer, others express mesothelin at much lower levels, in which case you might want to think about invoking some kind of more specific biomarker strategy to identify these patients, whether they're high expressing or low expressing. And as well, you could think about some kind of, combination diagnostic strategy to go along with that. So that would be kind of a more unique kind of approach to this. And it also kind of leads into a little bit more about why we are pursuing and why so many companies are pursuing this shielded approach. And so whether it's T cells you're using or NK cells, which I think may have a more muted safety profile, you want to think about, again, going after those tumors where the target may be more ubiquitously expressed. So for example, if you take EGFR, in addition to being expressed on tumor tissue, is also expressed on the heart. So you can imagine you probably don't want to take out that organ. So that's where some of these shielded approaches become very, very important.
Mm-hmm.
Did I answer your question?
Yeah. Anybody else want to jump in on that? I know, Nanna, I see your microphone's off. I don't know if you wanted to redirect or ask any questions around that.
No, I mean, I think Rachael raises a lot of very good points, and I think we see a lot of activity from multiple different formats going after a lot of these, tumor-associated antigens. And I think the trick will be to find the right format to balance getting a good, robust tumor-killing efficacy, but somehow avoiding the systemic or on-target but off-tumor toxicity with some of the initial approaches. I guess Elverum Therapeutics is a good example of a company that's trying a different novel approach to do that. I think they have a very intriguing technology of using bispecifics to engage only a subset of T cells, namely the gamma delta T cells, which act as a kind of bridge between the innate and the adaptive immune system, but then also having a tumor targeting moiety so that only tumors expressing or only cells expressing a particular surface antigen are targeted, and the gamma delta T cells are redirected specifically to target those. I think it remains to be seen, obviously, whether this particular cell type or NK cells or next generation CD3 engagers or whether a combination of these will be best suited. I think there's a tremendous amount of exciting clinical data coming out over the coming years, but I think already some of the data from Harpoon, from Amgen, and others in this area are showing that actually it is possible to use these sort of more next generation CD3 engagers to get early signs of activity.
Mm-hmm. Great.
If I could add just one point to that and agree with everything that has been said, what we haven't talked about is the non-tumor associated antigen targets like TCRs. And so that has actually been a really interesting proof of concept from a solid tumor standpoint. If you think about Immunocore and their recent successes in uveal melanoma, that also is the basis of the strategic discovery deal that we at Harpoon have with AbbVie, where they're providing TCRs and we are essentially providing the T cell engaging backbone of the TriTAC. And so we have high level of excitement and interest in seeing those progress as well as a different type of target, a solid tumor target for this type of technology.
Mm-hmm.
I think Elliot's probably best positioned of any of us to talk about Immunocore's success. But I think as an industry, we also have to ask, in terms of some of those technologies, how applicable are they going to be and how scalable? As you start to think about, we have a program for HPV positive cancers, but we are for HLA-A2 patients that express a certain HPV antigen. And so you're screening a lot of patients to be able to find those that might benefit. And so, what sort of differential efficacy does a program like that have to show versus other approaches to be viable? And then as an industry, how committed are we to, full profiling of patients so everybody's getting screened, because for individual companies that are smaller like ours to develop a CDX, it's not a cheap process, to say the least.
Mm-hmm. Yeah, and I certainly think the HLA restriction question is an important one, but alpha deltas was the first place to go. Immunocore, Ematics, I sit on the board of Ematics. I think that's exactly the right place to go. But they also are leading the way to the less restricted types, and as you know, there are several other HLA subtypes. And you think about the gamma delta TCR types, which are not restricted, and I think being able to demonstrate progress and is one side, and I think the other would be what Immunocore have done really well is focused, if you excuse the pun, but narrow it down to a disease type in which there are few or no other options for patients. Uveal melanoma is an excellent example of that, and so I think you can create the answer to the medical need in that sort of way. But notwithstanding, Kris, I agree. I think it is a subset of a subset of a subset is where you end up, and so really it's in those really hard to treat patients that the first class will come through. I think that's going to be important.
Mm-hmm. Yeah,
I think we also see a lot of companies taking the approach of if they have a T-cell engager or some other cell type, but targeting a tumor antigen or an ADC as well, could also be ADC approaches. If there is a tumor associated antigen which is very highly expressed in a small but well-defined patient population, that can be a really obvious initial proof of concept, and it may not be the big commercial opportunity, but showing clinical efficacy and showing signs of effect initially then makes it worthwhile to undertake the much broader screening exercise to get to the 10-12% of a much larger tumor type. I think mesothelioma is a good example of that, right?
And I would say, if I just add one more thing, Mike, is I think this is all centered around expression on immune cells, expression on of all different types on cancer cells, and you ultimately get down to some pieces of the dynamic in that microenvironment that no one really understands yet. So there's receptor turnover, there's copy number, there's what happens once the T cells kill the targeted the targeted cancer cell. Does another generation come up? And all of those questions we actually will answer collectively, because we all are doing a microcosm of experiment in that setting. And as data emerges, we'll see some things that do work in some settings and some things that don't. And hopefully the third generation of immune oncology will be, to use Rachael's expression, standing on the shoulders of us. We're not necessarily giants, of course, but as we go through as well, and there'll be successes and failures inevitably as we move to the next wave. And I think the answer may be not yet present for us, but we will know it soon enough.
Okay, so you touched on a bunch of different, interesting points and topics, in the last couple of minutes there. Heme versus solid tumors. How do we, access, more complicated tumor microenvironment? If that is indeed, a main barrier. Antigen selection, going beyond tumor-associated antigens, looking at intracellular antigens, neopeptides. We also talk about fit within standard of care. How do you deal with the paradigm of multi-drug treatment regimens? And also we talked about how do we make it more scalable and more accessible to a broader set of patients, particularly if you're going beyond the 10% of heme malignancies to the 90% of solid tumors in large markets. So maybe who would like to comment on that, and maybe Lisa, could you start a little bit of your thoughts beyond heme malignancies and solid tumors with your particular approaches?
Yeah, absolutely. So we very much are looking to move from heme malignancies into solid tumors. So, for example, our T-cell engager program, our CD20 by CD3, clearly is for heme malignancies, but we think that IgMs have a tremendous potential to really open up avenues within solid tumors. We have very nice data which shows, preclinically great efficacy in a host of solid tumor indications, again, really leveraging the strength of the IgM. And I think, as the panel has been saying, it's unlikely that one solution is going to solve everything. And it really is very much a situation where we have to iterate, and we all have to collectively learn from each other and build on that knowledge. But clearly, there's huge efforts, for the reasons you said, Mike, to move things like T-cell engagers into solid tumors. And that's because of the potential that we've seen in the heme malignancies, but because of the potential market opportunity that exists.
Mm-hmm. Anybody else want to touch on solid tumors?
Well, of course, it is ultimately the goal, as you say, Mike. It's 9 out of 10 cancer patients, and unfortunately, the number of cancer patients is growing, and so those numbers in absolute terms are going up and up. The other really interesting phenomenon is we're beginning to see new biologies emerge. So as the first generation of checkpoint inhibitors, so L1s, CTLA4s, and others, we see the tumors adapt. And so our lead program sits in the LAG-3 environment, and what's very clear is post-checkpoint inhibitor, you get a LAG-3 response. Now, LAG-3 exists biologically, but it's pronounced post-checkpoint. And so we're beginning to see needs that are emerging because of the treatment paradigm that patients are going through in first generation. And so not only are the numbers of patients going up, but also the patients who have used one option up already in terms of checkpoint then reemerge. And although the immune oncology paradigm has been transformational for patients over the last decade, it's really only transformational for about a quarter of patients with solid tumors. And the other 75% are still sitting out there using up options, getting longer and better quality life. There's no doubt about that. But I think we need to be very careful not to assume that the job's done either. We're really right at the beginning of this journey for patients, and I think for all of us here collectively, the intensity with which we look at that tumor microenvironment, the way in which you're looking at solid tumors, is also going to be colored by the other things that are emerging, and the increasing number of indications that have first line approvals means you get a wave of need in solid tumors beyond that. And I think bringing to bear cells, TCR type approaches, biologic approaches other than those that we're dealing with collectively is going to be very, very important into a huge.
And I might add to that and say that I think one of the things that's become clear in solid tumors is that a single target is not going to be enough. You have intra and inter tumor heterogeneity, and so solid tumors have very clearly shown us that even if you are able to successfully target an antigen or some other mutation, that they will mutate away from those. And so the notion of what's often called antigen spread is one that's absolutely critical for a therapy. And then as well, I think something that someone brought up earlier is, how do we think more and more about different combinations? And one of the challenges we have in our industry is novel combinations are very rare, right? But probably what's ultimately needed. And so, being able to think about innovative models for driving forward novel combinations are probably what's going to unlock this challenge that we're facing.
And actually, Kris, if I can just jump in there, one of the programs that IgM has in the clinic right now are DR5 agonist, which is in phase 1, all comers for solid tumors. We're actually looking to combine that with SMAC mimetic. So speaking directly to your point about novel combinations, and we're doing that very, very early on with the eye to what makes sense from a rational mechanism point of view scientifically. So both intrinsic and extrinsic cell death signals, seeing if that can't drive, greater tumor killing in a solid tumor setting. So completely agree with you on the importance of exploiting those types of novel combinations, but also to what Elliot said, that the challenges that we all face as drug developers, given the fact that there are a lot of options out there for patients. A lot of patients are in clinical trials. And when you're recruiting within the setting of a novel agent and a novel combination, you do tend to get really heavily pretreated patients who enroll in those studies, which is challenging in and of itself to show that your agent actually has the potential to overcome a host of things and challenges that we may not even be aware of as a collective industry yet.
Yeah. Lisa, I'm glad that you brought that up. I think those are two of what you and Kris both said are two of the things that weigh heavily on my mind in terms of the changing paradigm for oncology, and can our regulatory framework keep up with it, which is, this idea of novel combinations. And I know, organizations like the Parker Institute and others have done a lot to actually overcome some of the disincentives around doing that. But, they're one organization and it doesn't really kind of compensate for the entire system here. And that leaves it to organizations to start coming up with, and you guys have heard me say, the Frankenstein molecule, which is, if I can't combine with another novel molecule easily, can I just create one with more targets and more effector cells and try it and more co-stim domains and see what else I can put on it to try and do all those things at the same time. And that becomes a question of science and designing the right experiment to see if you're really testing what you think you're testing. But I think also very specific to oncology and immunotherapy is that our therapies rely on an intact immune system to provide effect. And what that may mean, and I think we're starting to see it emerge in at least some tumor types, is that moving patients that are earlier in lines of therapy may have a better chance of doing well with these therapies than those at the end of the line. And so that that really demonstrates that would be an upheaval to the way that we think about the development of medicine from a regulatory standpoint. And these are two areas that I think really need to be addressed by the industry, by FDA and other bodies to think about how we advance these medicines.
Yeah, we didn't get to the Frankenstein molecule topic soon enough, but Nanna, could we turn to the investor perspective? Maybe you could wrap up a little bit with what you've heard and your interest in. I didn't realize that Novo Ventures had invested in so many early stage platforms, but I'd love to hear your perspective.
Yeah, I mean, I'm impressed with the adventurousness or bravery of my fellow panelists here. I would say we're maybe not so adventurous. I'm not sure that we would invest in a company where the lead program was a novel combination. I think it's great to have novel biology, but it's also great to be able to test a new hypothesis fairly rigidly in the clinic. And I think we can only do that if we have grounds for comparison, some kind of existing benchmark that we can compare it to. And I think we're looking at a lot of known targets where there's some prior clinical validating data, but also some issues, and seeing whether any of these new formats would be well suited to address some of those shortcomings, but where we're not taking both format risk and target risk at the same time.
Mm-hmm. Great. And I hear that we can take an extra minute. Maybe Nanna, if you can maybe elaborate a little bit more on the investor perspective, any advice to give companies and things like that.
Sure. I mean, I would say I think immuno-oncology has been a super exciting area to follow for I guess all of us over the last decade or so. I think from an investor perspective, immuno-oncology is super challenging in terms of looking at preclinical data. I think sometimes we can get quite far with looking at human in vitro data, but I think I'm sure we can all agree that actually mouse models are poor and don't tell us a whole lot. And I think that's actually that's a big part of the reason why we're so interested in looking at programs that target cell types or targets that have some level of clinical validation, so that we have some kind of bearings of, what are the indications, what are the opportunities that we can address. So I'd say the main thing that we look for is some kind of previous clinical data, previous clinical understanding of the pathway, and then in vitro or in vivo data to underpin why this particular platform or this particular format is best suited to address that.
Mm-hmm.
And we
Great.
continue to be super excited about all the opportunities that we see in this area.
Great. Yeah, and it just seems like, it's a very interesting time for innovation. It's a great time for entrepreneurs to be in this space, and just the increasing amounts and sizes of Series A's, follow-ons, and IPOs that we've seen in the last year in this particular sector has been quite exciting. And so with that, I think I'm staring at 3 big red zeros, so I think we're out of time. But I thank the panelists for their contribution, and I think it was a very interesting, interactive discussion, and look forward to doing another panel on that Frankenstein molecule someday. So, all right. Well, thank you very much, and
thank you.
Have a good day.
Thank you.
Thank you.
Thanks. Bye-bye.